Wolbachia symbiosis. The genus Wolbachia is made up of several species of gram-negative bacteria. All species in this genus are obligate intracellular parasites or mutualists of higher animal species. These species have not yet been able to be cultured in lab without the presence of a host cell. They are a sister group to the genus Rickettsia, which includes pathogenic bacteria for diseases such as typhus and Rocky Mountain spotted fever. This group is considered one of the most common parasitic microbes and the most common reproductive parasite. They can infect many organs inside their host, but they mainly infect the ovaries or sperm, where they cause numerous types of reproductive and developmental changes in their host organism. And here is an example. This is a host cell right here, and then here is the Wolbachia colonies right here, and then the green in this part. Types of hosts. They are mainly found in arthropod hosts, and in the arthropods, they are mainly found in certain insect species. And this includes, but is not limited to, certain species of wasps, mosquitoes, fruit flies, and ants. Currently, we think that they infect 60 to 70% of all insect species, but that number could be higher with further research. They also infect other members of the arthropod group, which such as certain species of crustaceans and arachnoids as well. And the only other group that currently we know to have Wolbachia is certain nematode species. How are symbionts transferred from host to host? There are two ways for symbionts to be transferred from their host. The first one is horizontal transmission. This is where symbionts are transferred by environmental means. So something in the environment transfers the symbiont from host to host. This favors a more pathogenic or parasitic association. And in this one, multiple members of the same generation can be infected at the same time. The next type is vertical transmission. This is where the symbiont is transferred directly from parent to offspring of the host. And this favors a more neutral or mutualistic association. And this makes sense because the symbiont needs the host to reproduce to replicate itself. Transfer of Wolbachia and their host. They transfer mainly using vertical transmission, meaning they transfer from the mother to the offspring. So you have to have an infected mother to pass the bacteria down to the offspring. Having only an infected father does not work, does not pass down to any of the offspring. The bacteria are passed down only through the maternal line. While the bacteria is known to infect immature sperm and egg cells both, it is only found in mature egg cells, not in mature sperm cells. So, due to this, the bacteria manipulate their hosts to produce more females than males, since more females in a population would be more beneficial to them. And the four most common ways they do this is cytoplasmic incompatibility, parthenogenesis, feminization, and male killing. And these effects are beneficial to the bacteria and allow them to persist and be successful in the population. The first one, cytoplasm incompatibility, is the most common of the four techniques used. This is where bacteria induce non-viability in offspring of infected males and non-infected females, or females infected with a very different strain of the bacteria. It is thought that the bacteria in the testes induce alterations to sperm cells. A similar strain of the bacteria must be found in the egg cell to rescue the fertilized zygote from these modifications. If not, these modifications will interfere with early mitotic death or mitotic events and cause zygote death. The exact mechanism, however, is currently unknown. So this increases the fitness of females who are infected with the bacteria and also females who are infected with multiple strains of the bacteria. They have a higher chance of passing down their genes. Non-infected females have decreased fitness, and this allows the bacteria to pass through a host population quickly. This is seen in 8 different orders of arthropods, including butterflies, mites, beetles, isopods, ants, wasps, and grasshoppers. The next type is parthenogenesis. This is a form of reproduction without fertilization of an egg. These egg cells can develop into embryos and eventually adult organisms without the use of sperm cells. This results in all female offspring. Due to this, the bacteria have a better chance of surviving and populating since there's only female offspring. This occurs mainly in haplodiploid species. These are species where fertilized eggs, where they have 2 chromosomes, become female, and unfertilized eggs with 1 chromosome becomes male. The bacteria infect unfertilized cells with only 1 chromosome and induce the cell to double the chromosome, resulting in a female. This is seen in some wasp species, including parasitic ones, and certain mite species. The next type is feminization. This is a process where genetically male individuals are turned into females who can reproduce. The bacteria accomplish this by inhibiting the hormone androgen. This hormone is responsible for male characters such as the testes development. Without this hormone, male characters cannot develop, and female characters develop instead. This process leads to a sex ratio that favors the female. This is the less seen of the techniques described, and are only seen in 3 arthropod orders, such as crustaceans, isopods, and butterflies. These turned females are genetically still male with a ZZ chromosome, but are capable of female reproduction. In these species, new research is showing that the female chromosome, ZW, is being wiped out, and most females have ZZ chromosomes, indicating they were changed from male to female. Actually, in some populations of isopods, they no longer have a ZW chromosome at all. The final type is male killing. This is where the Wolbachia induce killing of genetically male individuals. This is similar to cytoplasm incompatibility in that both allow the bacteria to induce their own sex variation and recognize the host sex. This is seen in 4 different arthropod orders, including butterflies, beetles, flies, and pseudoscorpions. This is typically employed when the death of male siblings enhances the survival of their female sisters. Mainly seen in species with an already very high sibling competition level. In some ladybug species, there's such a high competition for survival between siblings that organisms produce cannibal larvae that eat most other larvae in the nest. The dead male larvae killed by the bacteria is food for the cannibal female larvae and favors their survival. Types of interactions. Since Wolbachia mainly reproduce by vertical transmission, we can infer that most interactions are then mutualistic or benign. However, this is mostly not the case. In arthropods, they are mainly considered a parasitic interaction. So even though the bacteria are considered parasitic, they usually don't cause an early death for the members, the arthropods. However, there has been seen to be one highly virulent strain of Wolbachia that kills pretty early on in the organism's life. And there also has been found in at least one species that the bacteria forms an obligate mutualistic symbiosis with the arthropod species. In nematodes, this is completely different. It's mainly mutualistic in nature. Most nematodes cannot survive without the bacteria, and the bacteria also boost the immune system of these worms. So the one obligate mutualism found in arthropods is found in a parasitic wasp species called Isobera tabida. And in this experiment, the scientists treated the parasitic wasps with antibiotics to kill the bacteria. And they found that in the wasps who were treated successfully, that the females had no egg cell production. And after further tests, they determined that the presence of the Wolbachia is needed for production of egg cells. And this was a photograph from their study. A right here is a control female who has lots of egg cell production, and then B is from a treated female with no egg cell production. The other unique case is the highly virulent strain of Wolbachia, and this was found in a certain species of fruit flies, and it considerably shortens the lifespan of the host due to massive reproduction in adults. And even though this strain was found in both larvae and adults, it did not start reproduction until the organism became an adult. The strain is inherited equally by males and females and was called popcorn based on its appearance in the bacterial cells in the host. We are currently unsure why it's so virulent because it does not appear to have any evolutionary benefits. And here's just a picture from that study of the host brain cells, and you can see how the bacteria just destroys the inside of the brain. So Wolbachia's usefulness in disease and the environment. Currently, there is great interest in this bacteria due to their potential in environmental pest control without the use of toxic pesticides. Also, understanding of their relationship to their host may help treat and stop the spread of multiple diseases, since many of their hosts are vectors for certain terrible diseases. Also, looking at how the bacteria manipulates the female line in hosts to control these organisms as well. The first type is dengue fever. This is a virus, and it's a highly deadly hemorrhagic fever infecting 50 to 100 million people a year. Currently, there's no treatment or vaccine available, and the only way to stop the spread is by controlling the vector. And the virus mainly spreads through the mosquito species Aedes aegypti. However, Aedes aegypti is normally not infected with Wolbachia bacteria. So in 2010, researchers wanted to find out if they put the Wolbachia inside these mosquitoes, if it inhibited the virus in the host. They found that the bacteria did inhibit viral replication and dissemination, and that these mosquitoes also had increased immunity and lifespans compared to the non-infected individuals. They also found that transmission of the virus was greatly reduced in infected mosquitoes compared to non-infected mosquitoes. However, they also tested another vector species for dengue fever, and they found that this species, Aedes albopictus, was not affected by the bacteria. The virus was not affected by the bacteria in the host. The other disease currently being looked at is elephantitis. This is a chronic lymphatic system infection caused by a nematode and causes extreme extremity swelling, and it's transmitted by multiple species of mosquitoes. Currently, however, it's being tested by using antibiotics as a treatment, and even though this would not kill the nematodes, it would kill the Wolbachia inside the nematodes, which would then allow the nematodes to die since they need the Wolbachia to survive. It also has been proposed to introduce male infected mosquitoes into the environment to control populations, since this combination of male positive Wolbachia and female negative Wolbachia would induce offspring who are non-viable.